Bismillahirrahmanirrahim. Welcome back, friends. Welcome back to another video lecture by Kami Microbiologist. So we are actually talking about the CBC, and in CBC, what we talked the CBC completely, which we mostly count it using hematology analyzer machine. But let's suppose if we do not have a hematology analyzer machine, or if we not afford it, so then we manually count this complete blood count. So we manually do that. So we actually talked the DLC, we talked the TLC, and we also talked the RBC count as well. Now if you miss these video lectures, so go back to my channel and watch these video lectures. So don't worry, I will also give the link in the description box, then you will check it. Now platelet count is again, this is this test of the CBC. It's actually mentioned in the CBC test. And if as we talked in CBC, as we talked in WBC count and RBC count and DLC and TLC, as we talked that we also we find out it using manual technique. In this case we also find the platelet count by manual method. Now what is actually platelet count test is? The platelet count test is, again, it is the blood test in which we count the number of platelet cell in the body. Then what are the main purposes of this platelet count test? In this test we diagnose to check out the too much bleeding purpose and the too much clotting purpose, that how the bleeding and clotting will be occur. Now this test can also tell us about the blood thickener. Let's suppose there are some medicine we are using due to which the blood are become thin, and due to this way what happen, there is less heart attack chances, because in the 50% what happen, the blood are too much thick. Due to this way there is a risk of the heart attack. So due to this platelet count test we can also check the blood thinner property. Due to platelet count test we can see the function of the platelet during complex surgery, like we have cardiac bypass and trauma surgery. Now let's see the normal range of the platelets. In male, female, we have the normal range of the platelet is 150,000 and to 450,000 platelet per microliter. In female the same ratio we have, but let's suppose during menstrual cycle the platelet count will be decreases. Okay, then we have the platelet. This is not the complete lecture of the platelet, but in this video we will just talk a little bit about the platelet to understand the platelet count test method. So let's talk that what is exactly platelet is. Platelet are also called thrombocyte, and it is the type of the blood cells which are smaller as compared to red blood cell and white blood cell, and it is colorless as well as non-nucleated. The diameter of the platelet is 2.5 micron. Now there are several shapes of the platelets. We have spherical, rod, and disc oval shape of the platelets as well. This shape of the platelet will be if it is in inactivated form. Now then we have dumbbell shape, comma shape, cigar shape, and some other shape of the platelet as well. Then we have the platelets, that what actually the platelets have. Platelet cells contain cell membrane, microtubules, and cytoplasm. Then we have the role of the platelets. Platelets are especially important in the blood clotting. So when the blood is clot, so what happen, there is no further blood loss from the body. Then we have the lifespan of the platelets. So the lifespan of the platelets is 10 days. It can also be 14 days as well, but mostly it is 10 days. Then this is actually the exact structure of the platelet under the electron microscope. Now this is the inactivated platelet structure, which is oval like shape or spherical in shape, and this is the activated form of the platelet. Now these things, as you can see, these are called filopodia shape. So this is the filopodia shape of the platelet, and this will be in activated form. Now this is actually the rough diagram of the platelets, that what is actually inside the platelet have. Inside the platelet we have alpha granule, we have open canicular system, dense granule, glycogen, microtubules and mitochondria. Mitochondria is the powerhouse of the cell. Microtubules can maintain the cell structure. Glycogen can store the food substances. Glycogen, dense granule and alpha granule, all these are storing the food substances and some other molecules inside the cell. Open canicular system is actually specially responsible to take the material inside the cell. So these are the main things that is present in the platelets. Then we have the parameters of the platelet count. In platelet counts, what are the most important parameters? We have mean platelet volume. In platelet we mostly check the mean platelet volume, we check the platelet crate, and we check the platelet distribution width. Inshallah ta'ala we will talk about each parameter one by one in detail in some other video lecture. Then what we required when we perform the platelet count? So first of all we required the blood sample in ADTA tube. Then we required the pipette, which is actually white blood cell pipette or RBC pipette. We just required this type of the pipette, as you can see, which contain a bulb and suction rubber like things. So this is the white blood cell or RBC pipette. We need a dilution fluid. This is actually platelet dilution fluid, not WBC dilution fluid. Okay, we required microscope, we required slides, hemocytometer slide, and then we required Leishman stain. Let's see the methods through which we can count the platelets. So we can count the platelet by manually as well as by automated hematology analyzer. Now using simple slide method and using hemocytometer method, how we can count the platelet. First of all, let's talk the simple slide method. In simple slide method, what we will first do, first of all we take the blood and we have to collect it into the ADTA tube. Now after that we put small drop of the blood, as you can see. Once we put small drop of the blood on the slide, then what we will do, we have to make this blood film or blood smear. Now how to make blood film or blood smear? We make it by putting first slide. This is the first slide. In the first slide we have to put the drop of the blood. After that we take another slide and we take it on the surface of the first slide, as you can see. Then we move it at backward. Now once we move it at backward direction, so what happened, it will touch the blood, as you can see. Once it touch the blood drop, so what happened, the blood will be spreaded, as you can see. Now what we will do after that, we have to push the slide or move the slide at forward direction. So what happened, it will make the blood it will spread the blood, and after that we get the blood smear. So this is the blood smear or blood film. It should be not more thick and thin, it will be normal, as you can see. Then after once the blood smear is formed, so what we will do after that, we have to cover it using Leishman or Giemsa stain. Now it should be stained using Leishman or Giemsa. Now once it is covered with Leishman or Giemsa for almost 2 to 5 minutes, after that we have to wash it using slow running tap water carefully. Okay, now once it is washed, so then it should be air dry, or using tissue paper it should be dry. After dry, what we get the slide in this form. So this is the slide with staining. Now after that what we will do, we have to count the cells at that portion. This is the tail portion of the blood film, this is the body portion of the blood film, and this is the head portion of the blood film. So blood at that portion, the platelet should be counted, not at this at the head and the body portion. Why? Because here the platelet are clumping with each other. Now once we see the slide under the light microscope, so we check the platelets. First of all we have to focus the slide at 40x. Once it is focused at 40x, after that once we see the blood cells, after that it should be visualized at 100x. At 100x we have to put means in this case we have to put what, we have to put oil immersion. Now after that, once we put the oil immersion, so we can easily visualize the platelets. As you can see, these small dots these small dots are called what? These small dots are the platelets. So we have to count it into different field. So we mostly count the platelet using 10 field. So this is, let's suppose, the first field, then we have to count it into the second field, third, fourth, till to 10 field. So this is how we can check the platelet or count the platelet in using simple slide method. Now using hemocytometer slide method, so how we count the platelet? First of all, we have to wrap our finger, as you can see in the picture. Now after that, we prick it. Once we prick it, so the blood will come out. This blood will be sucked using white blood cell pipette up to 0.5 mark. Then once it is sucked, so then it should be diluted using platelet dilution fluid, as you can see, up to the bulb area. After that, it should be loaded at this slide, means between the cover slip and the surface, the underlying surface. So once we load it at so after that, at 100x, mostly at 100x, what we found the squares, as you can see. These are actually the square which we count for the WBC. So we neglect these squares, and we have to focus on these square. As you can see, in these square we have to focus just 5 square. As you can see, this is the first square, second, third, fourth, and 5 square. We have to count the platelet and these 5 square. In these 5 square, it again, in this first square again contains 16 small more square. So the platelet should be counted in these 16 squares. So let's suppose if you count the platelets in each of the 16 square, it means that you count the platelet into 80 square, because if we multiply 16 with 5, so what we get? We get 80. It means that total we have 80 squares. Okay, so in 80 square we have to count the platelets. So this is, once we count the platelets, means so then how we calculate the platelet numbers? So this is the first method of the simple slide method through which we calculate the platelet. First of all, we have to write this is the formula of the platelet counting. How to calculate the platelet? First of all, we have to write the number of the platelets which we count in different field. Then we have to multiply it with the dilution factor, and we divide it with the field, number of the field. Mostly we count the platelets in 10 field. So these are the platelets, as you can see, counting of the platelet at a different field. So this is the first field, second, third, fourth. So at each of the field we count the platelets which are varied from each other. Once we count it, so then we have to add it. Then we multiply it with the dilution factor, which will be 15,000, and we have to divide it with the 10, because we count the platelet at 10 field. Now once we calculate it, what we get? We get 3,52,500 platelet, which is the normal range per microliter. Okay, now in hemocytometer, what we will do? We have to count the platelets in the 80 squares, as we talked, 80 square. Okay, now after once we count it, so how many? Let's suppose we get almost 235 platelets. We have to multiply it with the dilution factor, which is the 15,000. So what we get? We get 3,50,52,500 platelet per microliter. This is how to calculate the platelets. Now let's talk about the thrombocytopenia and thrombocytosis. Thrombocytopenia means that if the platelet level are decreases in the blood, we call it thrombocytopenia. Means if the platelet level are decreases from below 150, so then 1,50,000, so then we call that thrombocytopenia. Now how the thrombocytopenia will occur, or what are the main causes of the thrombocytopenia? Thrombocytopenia may be caused due to vitamin B12 and folate deficiency. These are the two most important substances which can maintain the platelets in the blood. Due to liver cirrhosis, because we better know that liver are specially responsible in the metabolism. So due to liver cirrhosis, the platelet are also decreases. Alcohol can also cause the destruction of the platelet. Now how? Because we better know that alcohol can cause the spleen to be enlarged, which we call as splenogamy. So due to the splenogamy, what happened? A large number of the blood will be stored. It means a large number of the platelet will be stored, because spleen is specially responsible to store the blood. It is the blood bank, that's why. Okay, once the blood are stored in the spleen, so what happen inside our body? The platelet level are decreases. That's why alcohol can cause the thrombocytopenia. Due to HIV or AIDS disease, or we have a virus which we call that Epstein-Barr virus. Epstein-Barr virus mostly attack on the blood cell, and HIV can also attack on the blood cell, especially white blood cell. Due to these viruses, the platelet level are also decreases. Then we have using chemotherapy and radiotherapy. Chemotherapy and radiotherapy, these two are what? These two are the special treatment which we mostly given to the cancer patient. So once we give these treatments, so what happen? It can also affect on the platelet, due to which the platelet count are decreases. Due to leukemia, the platelet are also decreases, which is the blood cancer. And due to myelodysplasia means bone marrow or blood cancer, in which the bone marrow secrete abnormal or immature blood cells. Now then we have thrombocytosis. So remember guys, if we have platelets more than 450,000, this is called thrombocytosis. So how the thrombocytosis will be occur? What are the main causes of the thrombocytosis? Thrombocytosis will be occur in the case of blood loss or bleeding, acute bleeding. In case of infection, how the bleeding will cause the means thrombocytosis, because if there is a high number of the bleeding, or if there is blood loss in a high rate, so what happen? The bone marrow will be activated, and it will secrete more and more blood, in which the platelets are in a large number. Due to this way, it can stop the bleeding process. That's why it can cause the thrombocytosis. Infection may also increase the thrombocytes. How? Because if there is infection, or bacterial infection, viral infection, what type of infection we have, so what happen? Bone marrow secrete white blood cell. So with the secretion of the white blood cell, we have also the platelet secretion as well, because bone marrow not only secrete the white blood cell, it can also secrete all blood cell types. So with all blood cell types, we have also the number of the platelets as well, increased number of the platelet as well. Due to iron deficiency, because iron maintain the platelet in the body. So if there is no iron or deficiency of the iron, so the platelet amount are also increasing. Spleen removal. Spleen, specially we talked that spleen are responsible to store the blood. So if the blood is not stored, so what happen? It will be remain inside the body. So what happen? There is a high rate of the platelets in the body. Then we have hemolytic anemia, in which the RBC are ruptured. Due to this way, only platelets are remaining. So what happen? We can say that thrombocytosis will occur. Rheumatoid arthritis, in which white blood cell attack on the joints or on the bones, mostly on the joints. Due to this way, the platelets are also increasing. Due to surgery and trauma, what happened? Again, in this case, bleeding will occur. So due to this way, the platelets are also increasing. Due to cancer, cancer can also cause the platelet increasing, because if there is bone marrow disorder, so what happen? It will secrete more and more blood cells. Due to this way, a large number of the platelet will also be secreted. So this is cause the thrombocytosis. So these are all about the platelet count test. I hope you guys get it about that. Thanks for watching.